So, a little bit of housekeeping before we get started. I am a little behind on grading just because I could not come into the school to get stuff. But by the end of the week, I plan to be caught up. So, hopefully, Thursday, Friday, I'll be able to give y'all a rundown of what I have, what I don't have, so you have 2 full weeks to get things started. I went ahead and did the project thing Monday. So, if you weren't here Monday, I can give you an idea of where I stand with your project. But today we are talking about the cell cycle. So, just like we have a life cycle, just like any living thing has a life cycle, single celled organisms and the cells in our bodies also have a life cycle. But before we can get into what that cell cycle looks like, we have to talk about chromosomes. Because chromosomes are a very, very critical, very, very important part of the cell cycle, we have to be able to duplicate our genetic information in order for genetic information to be passed from one cell to another. So, what is a chromosome? A chromosome is a tightly packaged bundle of DNA. Tightly packaged bundle of DNA. We need to keep this organized. So if you think of a specific gene as a Google Doc, our chromosomes are like our Google folders. They help us keep things organized. They help us find things when we need things. It's a lot easier to find a specific document that you are looking for if you keep it in a folder where you know where it is. Same thing with chromosomes. It's not just a bundled pile of spaghetti. Your DNA isn't just like in a pile in a clump to get all tangled. We keep it nice and tightly packaged in a chromosome. So, if you remember back to the very beginning of cells, we talked about the difference between prokaryotic cells and eukaryotic cells. Does anybody know the big major difference between prokaryotic cells and eukaryotic cells? We made a big Venn diagram. Yes. You got them flipped. But that's the big difference. Prokaryotic cells don't have membrane bound organelles. They don't have nuclei, they don't have endoplasmic reticulums, they don't have Golgi bodies. Prokaryotic cells don't have a nucleus. So if they don't have a nucleus, they don't have a place to store their DNA. So prokaryotic chromosomes have to look a little bit different because they don't have that protection of the nucleus to keep their DNA safe. So prokaryotic chromosomes. Like I said, we don't have a nucleus. So our chromosomes in prokaryotic cells, that's bacteria cells and the like, is stored in the cytosol. Prokaryotic chromosomes are stored in the cytosol. And most of them just have one circular chromosome. Well, most of them do. Some of them have a couple little extra small circles, but most of them, if you were to look at the DNA of any of the bacteria that live on the surface of our skin or in our guts, they will likely only have one chromosome. And instead of it being one big long string, it is in a circle, which makes it easier for them to replicate and gets it a lot less tangled. It's a lot easier to so if you have like those headbands or like a rubber band, those get like if you had a rubber band or a hair tie, or if you had just like your corded headphones, if you put them both in your pocket, odds are the circle is going to come out not having tangled up, but those headphones will come out a tangled mess. So prokaryotic cells don't have a nucleus to keep things secure, so they keep their DNA in a circle. But eukaryotic chromosomes, the chromosomes we have, are a little different. Remember, eukaryotic organisms are like you and me. We have our membrane-bound organelles, plants, animals, and a decent amount of other multicellular organisms have or are eukaryotes and therefore have these eukaryotic chromosomes. They contain much more DNA. Our DNA is a lot longer than the DNA of like a strep bacteria. We need more information, we need more blueprints than a single-celled organism would. We also have multiple chromosomes. So we'll talk a lot about how many chromosomes humans have compared to other organisms. We will talk about how chromosomes are inherited whenever we talk about genetics at the beginning of next semester, which is a lot of fun. But we have a lot more DNA stored in a lot more chromosomes, whereas our prokaryotic cells only had one. Our DNA is stored in the nucleus. DNA never leaves the nucleus. It never leaves it. It is far too important to be floating around the cell. Therefore, it is stored somewhere safe. Our DNA isn't in a circle, so we have to find a different way to store it. And what we do is we wrap it around special proteins called histones. DNA wraps around histone proteins. This keeps it from tangling up. So if you had one of those like corded headphone cases that you wrap your headphones around, or if you take your Chromebook charger and you wrap it around that little charging block, it's a lot less likely to be tangled up when you try to get it back out than if you were to just shove it in your backpack. So I'm going to draw it out for you. I would recommend you have some sort of doodle in your notes as well. The insert drawing function works on Google for those of you who have switched to Chromebooks. So we start out with some DNA, right? That DNA, I'm going to just turn into this line. This line's going to represent it. Will wrap around these little histone proteins. So DNA is going to wrap around that histone protein. Then a bunch of those histone proteins are going to join together. So now all of our DNA is wrapped around these proteins, and we've got a stronger kind of cord. Then these bundles, if this whole thing, I'll turn it blue, is a bundle, we'll represent it by one line, right? That will coil. If you've ever seen the old-timey telephones, that's what it looks like. Or those hair ties that look like weird springs or slinkies. And then that will supercoil. So it's that will supercoil. So the coils will coil. Until eventually that will form a chromosome. So DNA wraps around histones. Histones join together with other histones. Those big fibers made of histone proteins will coil. Those coils will coil, and those supercoils will condense into these little X shapes called chromosomes. Yes. Yep. Yep. Chromosomes are made of histone proteins and DNA, and that's pretty much it. So if we wanted to draw out a chromosome, we're going to label it. You can just draw lines. I like to draw squiggles just so I know that it's a bunch of DNA coiled onto each other. So it can be like that. You could draw it just like that. That would work fine. This whole thing is a duplicated chromosome. This whole thing is called a duplicated chromosome. It is made of two legs. So if you were to split it down the middle, and these are called sister chromatids. Sister chromatids. So these are in their duplicated form, which means that this leg of the chromosome and this leg of the chromosome contain the same genetic information. Because at some point during cell division, we're going to rip it apart, but we'll get there later. Probably one of the most important bits is that this centerpiece where they meet is called the centromere. Centromere. It's all one word. Centromere. So, chromosome looks like a little H, looks like a little X. It is made of sister chromatids put together and joined by a centromere. Anybody have any questions? No. All right. So that is chromosome structure. Now we get into the cell cycle. Cell cycle. During the cell cycle, a cell will grow, it will prepare for division, it will divide into two daughter cells, and each of those daughter cells will move into its own cycle. So just like a butterfly's life cycle is like larva, caterpillar, cocoon, butterfly, a cell's life cycle is grow, prep, divide, new cell, all over again. So during the cell cycle, a cell grows, prepares for division, and divides into what we said yesterday, 2 daughter cells. Each of which have their own cycle. So cell grows, gets too big because that surface area to volume ratio gets too small, just divides, prepares for it, duplicates its DNA, all that fun stuff. Once we've got everything we need to divide, we divide, and now we have 2 daughter cells. Those 2 daughter cells will grow until they get too big that it's time to divide. They'll prepare for division, get everything in order, divide into 2 new daughter cells, which will grow until they get too big. They will prepare for division, they will divide, they will have 2 daughter cells, and over and over and over and over again. Yeah, nothing too crazy. But just like prokaryotic and eukaryotic cells have different chromosome types, prokaryotic cells and eukaryotic cells have slightly different cell cycles. We'll focus more on the eukaryotic cell, but I want to briefly touch on the prokaryotic cell before we get too far into it. So, we're going to start with the prokaryotic cell cycle. Prokaryotic cell, that's really bad. Prokaryotic cell cycle. It has a specific pattern. This pattern is the same in almost all prokaryotic cells. It's not like different prokaryotic cells have different mechanisms of division. They all pretty much follow the same pattern. That is growth, DNA replication, cell division. Growth, DNA replication, cell division. They get too big, they decide to split into 2 cells, so they double their DNA. Each new cell gets a set of DNA. Continue. This happens rapidly under ideal conditions. I talk about strep throat a lot for examples, so I'm going to use it again. If a strep cell gets in the back of your throat, that is ideal conditions. It's got all the sugars that you eat. It's warm. It's damp. It loves it. So, ideal conditions. You get a couple strep bacteria in the back of your throat. Boom. By the end of the day, you feel kind of gross because that cell division will happen rapidly. But we don't know a lot about the details. When stuff on a cellular level happens really quickly, it's really hard to study. We don't know what triggers it. We don't know a lot of the chemicals that go into it. We don't know if there's an underlying pattern or if there's an underlying regulation system. We don't know a heck of a lot. There is always job security in biology. What we do know is when prokaryotes divide, it is a type of asexual reproduction. So, in prokaryotic cell division, asexual reproduction equals division And that is also known as binary fission. So if you've ever heard the term binary fission before, it's the same thing as prokaryotic cell division. It's the same thing as asexual reproduction. It leads to 2 genetically identical daughter cells. It leads to 2 genetically identical daughter cells. So if a strep cell were to divide via binary fission, if a yeast cell were to divide via asexual reproduction, the 2 cells that result from the first cell are genetically identical to the first cell. That is prokaryotic cell cycles. Anybody have any questions? All right. Moving on to the eukaryotic cell cycle. We will talk about the eukaryotic cell cycle for the first half of next semester. So this is really important. It will come back and come back and come back and come back. So the eukaryotic cell cycle. Eukaryotic cell cycle. We know a lot more about this than we do about the prokaryotic cell cycle, so much so that we have it broken down into 4 phases. The first phase is called G1. Usually G and then a little sub 1. But like G1 will do. In G1 phase, this is where most of the growing takes place. Technically, the G stands for gap, because when they first figured out the cell cycle, they thought it was pretty inert during this time. But as we've learned more about the cell, we know that cell growth takes a lot of effort. So they have changed the G from meaning gap to meaning growth. So first phase after in the cell cycle that we're going to talk about is G1. The next phase is S phase. So we've got the G1 phase and we've got the S phase. The S stands for synthesis, not photosynthesis, DNA synthesis. During S phase, DNA is replicated. Now we have 2 sets of identical DNA. So, G1 phase, S phase. The third phase is G2. G1, S, G2. In G2, this one is relatively short. Not as long as G1. Doesn't take as long as it would. But this is essentially just prep for division. We make the organelles and molecules we need. So both cells will need things like Golgi bodies. Both cells will need ribosomes. Both cells will need endoplasmic reticulum. So during G2, we during G2 we get all of our organelles in order. We make sure that we have enough to go around. Yes. Exactly. Last four, or the last step, M. M stands for mitosis. This is cell division and cytokinesis. So mitosis and cytokinesis. Cytokinesis is the process of that last snip between the two. This is cell division. After M phase, we will go right back to the beginning, and each one of those daughter cells will go straight into G1. But because scientists like to name things multiple times, if we took, ooh, that's not a letter. If we took G1 and S and G2, so all of the cell cycle except for mitosis, we call this interphase. Which will make a lot more vocabulary sense when we talk about mitosis tomorrow. So, if you haven't written anything down so far today, I need you to write down this next little chart because this will help you out immensely. So, we're going to start by just drawing a circle. It's going to be a little bit of a wonky circle, and I'm going to make a little gap like that. I'm going to make another circle on the outside. So I can write in the circle. It's a little smooshed, but it'll be all right. Then I'm going to make some arrows. So we know which direction we're going. This is going to represent our cell cycle. This little arrow right here, I will even change colors. This little arrow right here is mitosis. And this big arrow is interphase. And we're going to divide it up. So, roughly find the center. It doesn't have to be perfect. And we're going to divide it up. So, we're going to have a line going from the beginning of this arrow to the circle. So, we can take mitosis out as a chunk. And interphase is divided into 3 parts. So, we're going to divide this other chunk into 3 parts. Just like that. 4 phases of the cell cycle. 1, 2, 3, 4. So, right after mitosis, we have G1 phase. This is growth. Okay. After G1, we go into S phase. S phase is DNA synthesis. Last phase in interphase is G2 phase. And we're going to call that prep. G1, S, G2. We're growing, we'll duplicate our DNA, and we're going to prepare for division. Now, mitosis is also known as M phase. Also known as M phase. And it's divided into two parts. Those parts are mitosis and cytokinesis. This is our cell cycle. We will be acting within this kind of frame of reference for a very, very long time. So, if you have any questions, yeah. Meiosis is division, but with sex cells. So we've got like a whole nother process, which we will talk about. I love meiosis. Right now, this is run-of-the-mill cell division. You get a cut on your hand and you need to make more skin cells. Bacteria replicating, or not bacteria, because those are prokaryotes, but like single-celled organisms replicating. Just like your body growing in general, that kind of stuff. But yeah, this is a cell cycle. Every eukaryotic cell does this. G1, S, G2, M. Over and over and over again. Just like we get like larva, caterpillar, cocoon, butterfly. Just like that life cycle, but with cells. If you have any questions, please let me know. I love this stuff. There's an exit ticket on Canvas for you to fill out when you got the time, which you definitely do. So, yeah. Nothing too crazy. Tomorrow we talk more in depth about the phases of mitosis, and that will kind of take us through Friday and Monday, I believe. Yep.